So I'll start the meet. Hello and a very good morning to everyone, and good evening to two of our speakers from abroad who are virtually present to this international webinar on this current research trends in biological science. It's my great privilege and pleasure to welcome you all on behalf of the Department of Botany, Chuchampur Government College, Lamka, Manipur, India. And myself, Chinwan Thangwite, Assistant Professor, Department of Botany, Chuchampur Government College, Manipur, India, coordinator for this international webinar. Those of you who have joined across the globe, welcome to this webinar. And this webinar, it's hosted by Department of Botany, Chuchampur Government College, Lamka, Manipur, India, in collaboration with Penglam Kuak, Lamka, Manipur, India. So before starting, I'd like to request to all the participants to kindly mute your audio or your voice. There will be a Q&A for 10 minutes after each speaker give the presentation. The purpose of this webinar is to acquire the science fraternity about recent advancement in multidisciplinary fields of biological science, and to discuss an overall perspective on the latest results in this field. Hope we'll have a successful and informative webinar today. Luckily today we are very happy that we'll be having 3 guest speakers. 2 are from abroad and 1 from India. So I'm going to introduce each speaker very briefly. The first speaker will be Sir Dr. Pau Biak Sang from USA, and he will be delivering a lecture from 9:10 to 9:40 a.m. And after he give the presentation, 10 minutes will be given for Q&A, and after that one we'll proceed to the next speaker. The second speaker will be Sir Kumbialal Hausel from USA. He'll be giving a lecture from 9:50 to 10:20 a.m. And the third speaker will be Sir Joshua Khumlianlal from India from 10:30 to 11 a.m. I want to request Sir Dr. Pau Biak Sang. Sir Dr. Pau Biak Sang, could you please unmute your voice?
Hello, can you hear me?
Yeah, I can hear you, sir. Okay. So could you please present your slide? Okay. Are you ready, sir?
Yes.
So I'm going to introduce our first speaker, Sir Dr. Pau Biak Sang. And this Sir Dr. Pau Biak Sang, he's a molecular biologist, biochemist, and microbiologist. And he completed his Bachelor of Science in Microbiology specialization in the year 2007 from Delhi University, a Master of Science in Biology in the year 2009 from Indian Institute of Science, Bangalore, and Doctor of Philosophy, that is PhD, in the year 2014 from Indian Institute of Science, Bangalore. And his thesis topic is DNA repair protein in mycobacteria. Apart from that, he was a postdoctoral research fellow from the French National Center for Scientific Research, that is CNRS, Montpellier area, France, from 2015 to 2017. There, he worked on a project related to DNA replication origin in human. Apart from that, he was also a postdoctoral research associate from the University of Texas Health Science Center at Tyler from March 2017 to March 2020, Tyler, Texas area, United States of America. There he worked on a chromatin remodeling complex, its role in cancer and DNA damage response. Currently, he is a postdoctoral research from Department of Cancer Biology, Gardner Bernardin Cancer Center, Loyola University, Chicago, Illinois, United States. He has published more than 13 research articles in internationally acclaimed journals and also articles to his credit. So he will be delivering a lecture on the topic DNA damage and cancer. So I hope this talk will enrich the student and all of us alike. Over to you, sir, Dr. Pau Biak Sang.
Okay, can you hear me?
Yeah, I can hear you, sir. It's audible.
Okay, I'll start now. Thank you very much, Mohan, for the introduction. So today I'm going to talk on DNA damage and cancer. So since we have audience from maybe from non-biology background and also from different qualification, so I'll make this. I have made this presentation very simple. So to get started, as we all know that the human body is made up of different type of cells, and the human body is made up of millions of these different type of cells. Although they are all different, they all undergo a process of cell division and multiplication. So their somatic cell division is through mitotic. Mitotic is known as mitotic cell divisions, and they go through different phases, and in which a single cell is divided into two, and then two cells will become. So we all know that there are different type of cells, and they go through cell division and multiplications. So every cells undergo division and multiplication, and so one cells become two, two to four, four to eight, and so on. So this is the cell cycle which every cell goes through, and there is a growth phase, synthesis phase, and another growth phase. So during the DNA synthesis, DNA is replicated into two. DNA is double, so that every daughter cells can get the same copy of genes from the previous cells. Cell cycle is a regulated process, as I have said, through cyclin-dependent kinases. So different cyclins are activated in the different phase of the cell cycle, and so they play a very important role for the cell proliferation, because if there is any kind of inactivation of cell cycle, then the cell will stop growing. But cell inactivation is also important, because when the cells are not in a good condition, like there's a DNA damage, or if there is a lack of growth factors, or if there is a problem with DNA replication, then the cell needs to stop for a while. So for that, there's a checkpoint in every phase of the cell cycle. And how this checkpoint works is that it is through through a series of pathway. Uh, so let's say, for example, if there is a DNA damage, then there is a checkpoint, different kinases like ATM, ATR are activated, which will activate phosphorylate like a protein called P53. So P53 will phosphorylate P21, and this will inhibit this G1 cyclin-dependent kinases. So once this cyclin-dependent kinases is inhibited, then it will not go through the second phase. So this is a kind of a checkpoint which cells have, so that it will stop the cells for a while, and then allow the cell to maybe go through different pathway. For example, if when there is a DNA damage, then there is a phosphorylation of P53 through ATM or ATR. And what P53 does is that it can like arrest the cell cycle. It can stop the cell cycle, or it can stop the cell cycle for a while, so that the DNA damage can be repaired so that the cell is healthy. Or if there's a lot of damages in the DNA or in the cells, then the cell can undergo apoptosis. Apoptosis is a kind of programmed cell death, which is very normal for the cells. So apoptosis, or the cell can undergo senescence. Senescence is like the cell will stop growing and remain in an inactive state. So any defect in any of these proteins which are important for the cell cycle regulation can lead to abnormal growth of the cells. It can lead to abnormal proliferation of the cells, and it can lead to cancer. Initially, people thought that there is a defect in these proteins which lead to cancer, but it is not. It is not the protein which are defective, but later on people realized that it is not the protein, but it is the DNA which is defective. So let me go back a little bit about DNA. We all know that the human cells has a nucleus which contains the human chromosomes, and chromosomes is basically a very tightly packed form of DNA. So DNA is made up of four bases: adenine, guanine, cytosine, and thymine. And as you all know that, as biologists will know that from the DNA, through a process known as transcription, it forms RNA, and from RNA, a protein is formed. So this protein is the protein which are present in all the cells, like the cyclin-dependent kinases we are talking about, the P53, or all the proteins are made from the DNA. So the genomic integrity of a human cells is very important to have a healthy cells and to have a protein which is functional. So maintaining the genomic integrity of the human DNA is very important. Yes, we are saying that it's very important, but still, the DNA, the human DNA are subjected to a number of damages due to like maybe UV rays or due to ionizing radiation or due to the cellular DNA replication process itself, like the DNA polymerases, there can be errors in the cell itself or due to like reactive oxygen species which is generated from mitochondria. So the human cells are actually constantly damaged every day. There are thousands of DNA damages every day. So these are the type of damages which can happen in the cells, in the DNA, like the base mismatch or base mutations, or there can be a single strand break or double strand break or DNA cross-linking. But unfortunately, we have a DNA repair system in the cells also, which will repair the damage in the DNA. So there are different kinds of DNA repair system in the human body, like the mismatch repair system, base excision repair system, non-homologous enjoining, and nucleotide excision repair, etc. So whenever there's a damage to the DNA, this repair system will get activated and they will start repairing the DNA. So these are the different pathways which I just talked about. The base excision repair pathway basically repairs a single bases. Nucleotide excision repair pathway repairs like more severe kind of damages, and also there are homologous recombination and non-homologous enjoining. I'll not go through the details of this repair pathway, but I just want to let you know that the cell has a system to repair whenever there is a DNA damage. But again, these repair proteins which are involved again are made from DNA. So if there is a damage in the gene of these repair proteins, then these DNA repair proteins might be defective and they will not be able to repair the DNA. So this will lead to mutations in the DNA. So one example of mutations due to damage in the DNA is like deaminations of cytosine. So as you know that DNA has like A, T, G, and C. So uracil is not a common basis present in DNA, but due to deaminations of cytosine, it will generate uracil. C will be converted to U, and if this U is not repaired by the base excision repair pathway, then it will get replicated and it will the DNA polymerase will tend to add adenine against U, and then when the DNA further replicates, then it will form AT. So the base which was initially GC will turn to AT. Similarly, there is whenever there is oxidations of guanine, if 8-oxo-G is not repaired by by DNA glycosylases, then it will get passed on, and then during replication, it will be G will turn to T. So this is the kind of this is how mutations get generated when there is a defective in the DNA. So these are the different types of mutations which is present in the cells. So gene mutations have like there are different kind of point mutations. So for example, point mutations can be either silent, nonsense, missense, etc. So silent mutation is where the DNA mutation does not change the protein amino acid. But in a nonsense mutation, the mutation will change into a stop codon, and it will lead to stopping of the protein synthesis, and it will lead to formations of truncated proteins. Missense mutation is a mutation where a mutation will change the amino acid. So this will be a single amino acid change in the protein. So most of the time, single amino acid change might not have a very drastic effect, but if the mutation happens to be in the critical region, let's say the active site region of the enzyme, then it can be defective. So there are also many diseases which is due to single amino acid mutations. Also, a more severe kind of damages like DNA double-stranded break and all can lead to more chromosome mutations. So chromosome mutation can be deletions in the chromosomes or duplications of a region of chromosomes and inversions, etc. So this is how mutations get generated in the human body. So when mutations happens in some of the genes which are controlling the cell cycle, so this is the normal cell cycle. We have a gene, and this gene generate the protein, and this protein is functioning normally. But when the genes are mutated, then these proteins happens to be functioning in the cell cycle process. Then there can be loss of function mutation, or there can be gain of function mutation, which will lead to abnormal cell cycle, and it lead to cancer formations of cancer cells. Cancer cell is nothing but abnormal growth due to abnormal cell cycle processes. So this is how a cancer is formed. So again, this is another slide where a normal cells has a DNA mutations which are functioning in the very important important part of the human cell cycle, and this will get mutated, and then this will mutation will pass on, and it will lead to uncontrolled proliferations of these cancer cells. So so these are the different carcinogens around us. Carcinogens are actually mutagens, or any factors which cause mutations. And when the mutations leads to cancer, it is also known as carcinogens. So these are the different type of carcinogens, like tobacco chewing, or different industrial wastes, or viruses itself can cause mutations in the DNA, and it can lead to cancer. So cancer usually, as I've said before, that mutations always happens in the cells, and cancer, it does not always give rise to cancer. Usually cancer happens when there's a mutation in the cancer critical genes, and these are the genes whose mutations can contribute to development of cancer directly. So they are divided into two. One is oncogenes and the tumor suppressor genes. Oncogenes are genes where there's a gain of function mutations in the cells, which will drive the cell toward cancer. So they are also known as proto-oncogenes. And this gain of function mutation will lead to cell proliferation. They are usually, examples are the growth factors, growth factor receptors, or signal transductions like RAS or EGF, etc. So when there is a gain of function mutation of these growth factors, then it will lead to uncontrolled growth of the cells. And tumor suppressor genes are genes which, if there's a loss of function mutations, will lead to increase in the formations of cancer. They actually usually inhibit cell proliferation. Maybe they are playing important role in checkpoint kinases, and once they lost their function, then they will lead to more proliferation. Another group of genes are the genome maintenance genes. These genes are like the DNA repair genes we talked about, all the proteins which are functioning in the DNA repair process. If they have a mutation which lead to loss of function, then they will not be able to repair the DNA, and then it will lead to more and more mutations, and it can lead to cancer. So people have sequenced the genome sequence of cancer from cancer patients, and they have found that there are some genes, like most of them are the cancer critical genes, as I have talked about, like the P53. P53 is one of the most highly mutated genes in the cancer. So this is the number of the percentage of P53 mutations in different type of cancers, like colorectal cancers. It is highly mutated, like 12% of all mutations is due to P53 mutations, and also the other breast cancer or neck cancer. So every cancer has, most of the cancer has mutations in P53. Although they could also have multiple mutations in other proteins, like the RAS pathway or other pathway, but this is just an example of how mutations cause cancers. So this is the protein region. This is the DNA region where the mutation occurs. So there are several amino acids which are mostly the hotspot for mutations in P53. So P53, again, as I have said before, it is important, very important in the cell cycle process. It has a multiple role in the cells. So whenever the DNA damage occurs, then p53 can arrest the cell cycle or leads to apoptosis, etc. But when the p53 is defective, then it will not be able to function properly, and it will lead to abnormal growth of the cells. So these are also other different pathways. Most of them are like the signaling pathway, which so they are also known as oncogenes, because if there is a gain of function mutation in any of these signaling pathway, like the RAS pathway or the Wnt signaling pathway, then it will lead to more growth of the cells, because this pathway is important for the cellular growth and survival. But if there's mutations or hyperactivation, gain of function mutations of these any of these proteins, then it will lead to more proliferation and growth of cells, and which will lead to cancers. So as I've said before, tumors are formed uh, in a cells which is normal, but where there is a more mutations, in which there is an inactivations of tumor suppressor genes, which will lead to more cellular proliferation. And also one characteristic of a tumor is that they have genome instability, which means that they will lead to more and more mutations, and which will lead to more proliferations of cells, and which will lead to cancers. So here are some of the characteristics of cancer cells. So they sustain proliferative signaling, they evade growth suppressors, so they also resist cell death, like the apoptosis or senescence, they resist that, and they induce angiogenesis. Angiogenesis is like formations of more blood vessels, and they activate invasions and metastasis, which means they can go through the bloodstream and then go to different areas of the body also. And they usually have deregulated cellular energetics and metabolism. They usually have very high metabolism because they require more energy for growth. So they have deregulated, and also they avoid immune destruction, because some cells are usually destroyed by the immune system, but these cancer cells, they have the ability to avoid immune destructions. And they generally have genome instability, which means that the genome is very instable, and they can be more and more damages, which lead to cancer heterozygosity, and there can be a lot of different cells, which lead to which make the cancer cells more difficult to treat. So going on to the kind of treatment which is available for cancers, there are three types which are basically the main kind of treatment used. Surgery is like the removal of the cancer from the body. These are useful for solid tumor which are localized in one places, usually like the benign tumor, which does not metastasize, which does not invade other tissues. And the second is like radiotherapy, where people use a high radiation of energy, high dose of radiation to kill the cancer cells. But most of the time, it can also affect the neighboring areas. So nowadays, there's a development in this field also, like where they precisely target only the cancer cells. Like there's something called a cyber knife, which will help in targeting a very precise location to prevent the side effect. The third one is chemotherapy. Chemotherapy use drugs to kill cancer cells. So, but they usually kills cancer cells which are fast growing. But the human cells also have some of the cells which are also fast growing, like the hair follicles. So which will lead to a lot of side effects from the chemotherapy treatment. So nowadays there are also people are doing more personalized treatment. So personalized treatment means based on the person's need. So what they usually try to employ synthetic lethality for cancer treatment. Synthetic lethality means that if they can inhibit two genes which are working in the same pathway. Let's say this is the normal cells, and this is the cancer cells which has a mutations in gene A. And then if you manage to inhibit gene B, because gene A is already defective, And if you can defect can inhibit gene B also, then it will be lethal for the cancer cells. That means that the cell will undergo, the cell will die, or the cell will go through a different pathway like apoptosis. So another way is like to use drugs, a combination of drugs. So if you in these cancer cells, if you just kill one protein, then it will still lead, the cell will be still viable because there is a pathway B. But if you manage to kill both the pathway by using combination of drugs, then it might be lethal for the cells. So this is another example of synthetic lethality, where this is a normal cell which have the normal base exon repair pathway, like the homologous recombination and the base exon repair. But in the cancer cells, let's say there is a defective in the homologous recombination, and this will lead to cancer. But so if you sequence the genome of this patient, and if you know that, okay, this patient is BRCA mutated, then you already know that this patient is BRCA mutated, so you may be able to inhibit another protein which is PARP, which is functioning in the base exon repair pathway. So when you make more defective in the cells, then the cell will not be able to tolerate the genome instability, and it will die. So this is a kind of nowadays people are doing more of a personalized treatment by first sequencing the gene of the tumor. They will take the biopsy, and then they will sequence, and they will know that, okay, this person has defective in this particular gene. So if we treat in this way, then they will be able to treat more specifically. So this is some so this is a figure which I took from one company who are working on this personalized treatment. In this one, this is the cell cycle which I've shown here, and then this cancer cell has defective in the ATM and P53. Now they have generated a drug which will inhibit the CHEK1 and CDC. So if you can inhibit CHEK1 and CDC in these cell lines, then maybe the cell will be more, the cell will die. So this cancer cell will die more easily. So this is how people are making a more of personalized medicine nowadays. So to summarize my talk, cancer is a disease in which abnormal cells divide uncontrollably and destroy body tissues. And cancer is basically caused by mutations in cancer critical genes like the tumor suppressor genes or the oncogenes. And several environmental, lifestyle, chemical radiations can cause mutations in the cancers. So we can't exactly prevent ourselves from having a cancer because we are always exposed to UV radiations or radiations all the time. And also the human cellular process itself is defective sometimes. So it is very difficult to prevent ourselves from cancer, apart from like having a healthy lifestyle and always putting sunscreen to prevent from UV or to prevent exposure to chemicals which are carcinogens, etc. So as I've said, cancer-causing agents are known as carcinogens. So it can be anything which can cause cancers. Recently, in USA, they found out that the baby powder from Johnson & Johnson is carcinogenic. So they stopped the uses of baby powder like that. So also nowadays, personalized therapy through synthetic lethality can help in more specific cancer treatment. Thank you for your time, and I can have any questions.
Yeah. Thank you, sir. Thanks for your presentation. The first question is that from Zenhoim 1. She want to know what is the abbreviation of EGF?
Oh, that is epidermal growth factor.
Okay.
So they are like the oncogenes, they are the growth factors, and if there is a mutation, gain of function mutation, then it will lead to more growth of the epidermal cells, and it will lead to skin cancer.
Okay.
Yeah.
Another question from Nombaite. So she's asking, is it within our control to prevent or reduce intensity of damage of DNA?
Yeah, like I've said in the last slide, the way to prevent damage of DNA is to have a healthy lifestyle, like to avoid alcohol or smoking, because it is known that smoking can cause more damage to DNA, and it can lead to lung cancer. So to have a healthy lifestyle, and also to prevent exposure from anything which is carcinogenic. So that's the only way we can do. And also, yeah, like I said, like to use like maybe sunscreen to prevent from UV. Although we can't do all the time, but these are the ways we can prevent DNA damage. Yeah.
So another question from Nombaite. She is asking, can we improve our genetic composition through lifestyle?
Genetic composition is always the same, but yes, there is nowadays, not nowadays, there is also epigenetic factors which I didn't talk about here, which can lead to cancer, like methylation, which are epigenetic, which is due to which comes after our birth. So yeah, we may be able to prove the epigenetic factor of our DNA, but the DNA itself can't be changed. But yeah, we can change the epigenetic factors by having a healthy lifestyle.
Okay, so there is another question. So what are the P53, P60, etc. in our body? I think in our body. So what is the main
Yeah,
role?
so they are proteins. They regulate the cell cycle, as I have shown in the slide. So they are a very important pathway. They are proteins. They are transcription factors. Most of like they regulate transcription of other genes. So they are okay. Let me go back to one slide here.
Is it synthetic or is it nature? Can we?
It is a protein. It is a protein. It is natural protein which is present in the DNA, and it helps in the tight regulation of the cell cycle. So once if the P53 is defective, then it can lead to a defective DNA repair, defective cell cycle arrest. So the cell cycle will not be arrested, and it will lead to more growth, more growth means cancer. So these are proteins. P53 is a protein. Yeah. Short answer is P53 is a protein.
Okay. Another question. Now what about P60? Is it there any P60?
Yeah, they are named based on mostly on the molecular weight of the protein. Let's say P21 has molecular weight of 21. P60 I'm not aware of, but maybe because there are so many proteins in the human body which I may be not aware of. So I don't know whether she knows there's a P60. But
Okay.
yeah, yeah.
So another question from Tem Zenhoimwan. How come Johnson baby powder is carcinogenic? Why it is not banned in India?
I don't know why it is not banned in India, but maybe it is already banned in USA. Yeah,
Okay.
it's carcinogenic because it has some compounds. I think I'm not sure which compound is that, but yeah, I can look up in the internet what is the compound which is carcinogenic in Johnson baby powder, and it is better not to use that anymore, I think.
Okay. In our place, we still use now. When somebody got a new baby, we used to give those.
Yeah, yeah.
So another question more, sir. So from Roslyn Nasia, what is your
Yes.
PhD work related to, and what are you working on now?
Oh, my PhD work is related on DNA damage in mycobacteria.
Hmm.
Right now, I'm working on cancers. I'm working on telomeres. The ends of the DNA are called telomeres, so I'm working on telomeres. Yeah.
Okay. So another question more. How do smoking, tobacco, etc. break the DNA cells to cause cancer?
So tobacco smoking, there are a lot of chemicals in the smoke. So once when you smoke, and that the fumes get accumulated in your lungs, so it will form a tar, it will be a very black tar formation, and this tar is a chemical, and it can damage the DNA. So if you look at like some images which is surfacing in the internet, you might see that the difference between smoking lungs and non-smoking lungs, and smoking lungs are like very black. So that is due to accumulations of chemicals from the fumes, and it can cause lung cancer.
So from Roslyn Nasia again, when you say about personalized treatment in relation to cancer treatment, can you elaborate it?
So what they do is they took the biopsy from the cancer patient, they sequenced the DNA of the cancer, and they found that which gene is mutated. So once they found that a particular gene which is involved in a particular pathway is mutated, then they can make a drug which is inhibiting a parallel pathway. Means, let's say if there are two pathways which is important for the cells, then they will try to make a drug which will inhibit the other pathway, and then it will lead to synthetic lethality. Synthetic lethality means like some pathway, some process in the cells require two process, and if one is already mutated, then you can now mutate or what do you call it, destroy the other pathway, and it will lead to like this one. If there is gene A which is already mutated and you can like inhibit gene B, then it will be lethal. The cell will not be viable anymore. So if you know that what is the gene which is mutated in that patient, now you can treat gene B. So every cancers will not have this mutation. So this person will only have this mutation. So this is a very personalized way of treating the cancer.
Okay, so thank you. Another question from Gin Hong Sai. Is all cancer causing factor preventable?
Like I said, it's not preventable. You can't prevent cancer. That's why we have a lot of cancer. And also a person who have very healthy life, even that person can have cancer, because the human cells is not perfect, and humans are not perfect. So the cells are also not perfect. So there can be DNA polymerase which is defective. So it will lead to DNA replication which is defective. So it is not completely preventable, but to some extent, having a healthy lifestyle will prevent to some extent. That's what I can say. Yeah.
So another question from Zenhoi Muan. What makes Johnson baby powder carcinogenic? What are the chemical composition which make Johnson baby powder carcinogenic?
Yeah, I think I have already answered this question. I didn't remember the exact chemical, so one can look up in the internet. Maybe why Johnson baby powder is carcinogenic. Yeah.
Okay, sir. So from Moi Kulai, what is ATR? ATR.
Oh, ATR is it is a abbreviation for a long name called ataxia telangiectasia something, which is a kind of a disease. When ATR is mutated, then they will have a kind of a disease which is known as ataxia telangiectasia.
So from Moon Kim Singson, about the T cells, is this specifically for the brain or prone to other organs in our body? Tumor cells.
What I have presented is like a general. So as I've said, humans have different type of cells, and tumors can arise from any type of cells. So if you have brain cells, it will be a brain tumor. If you have it arise from skin cells, it will be skin cancer. So it can arise from anywhere of the cells.
Okay, thank you, sir. So from Juliet Kim, can dry vegetable locally made for future consumption cause cancer? Dry vegetable is locally made.
Mostly it should not, but yeah, there is no research because whatever people claim something, they do some research, and then they check whether the green or dry is more cancerous or not. So I don't think there is any published research on dry vegetables, so I don't know the answer for that.
Okay, the last question from Luni Munsong. The stages in cancer, what do they really mean by different stages? And According to your opinion, is Human Genome Project good or bad, and why?
Okay. So different stages means like initially they will have a very less mutation. The cancer is like very benign, and then it will have more mutations, or it will be able to metastasize. That means they can invade other part of the body. So that is like a different, the more advanced stage of cancer. So that is how people define the different stages of cancer. And Human Genome Project is very beneficial, very good, because not that this personalized medicine I'm talking about, and also in the field of research, if you know the genome of the cancer, then it will be very much easier to treat the cancer. And although it's still expensive to sequence the person's genome, but still it is very beneficial project.
Okay, I think I will wind up on the first session. Thank you so much,
Yeah,
sir.
no problem. Thank you. Yeah.